Okay, good morning, students. Today we are going to start the next lecture of crop improvement. That is all about kharif crops. And today's topic is breeding procedures for quality. Actually, here are two quality when we talk for, then it can be either protein quality or it can be either oil quality. But I will be putting stress only on the breeding procedures for quality, and quality when I say I will be discussing the procedures that are only related to protein quality, right? So these all are the contents that I'm going to discuss today. That is very first I will make you familiar what actually is quality, what must be the characteristics of quality, from where quality traits they can be obtained, what about breeding approaches, and then we will discuss genetic engineering. Since your topic is breeding procedures, that is including conventional as well as modern approaches. Conventional approaches is all focused on breeding approaches that is involving the certain aspects of breeding. But when we say modern technology, then number of times I have repeated, when we say modern technology, then what all we are going to discuss is all about genetic engineering, or say about the recent technology. Then while one means while breeder is going for breeding in terms of quality, then there are certain parameters which are to be considered by the breeder. So these are the important considerations which are to be kept into mind while going for the breeding of quality, breeding for quality. And there are often certain limitations of breeding. It is not like when you go for breeding in terms of quality, then all the results are going to be in the positive direction only. There are sometimes disadvantages or negative parameters often available, and we will also discuss about those parameters. So, introduction. I will be relating here two breeding procedures. One is for yield and one breeding procedure is for quality. If you could see your syllabus, then there are modern and breeding approaches in terms of, say, yield, and then you have one topic after the biotic and abiotic resistance, that is breeding procedures for the quality. But you know what is the chief, before I actually start with the breeding, the number of times, and in the very first lecture I discussed like the very chief, immediate and long-term objective of plant breeding is lying only in the increasing the productivity. If I don't say the term productivity, then the objective, even in the future, is only focused on the yield, so that all the food requirement of the people can be met. So objective of breeding is very clear. We have often discussed this in objectives when we were discussing a particular topic, that is objectives of the plant breeding. At that particular time also I told you like the very chief immediate, that means the very immediate requirement. And even if you talk for the long aspect, then even in the long term aspects, the one and only the one, the main objective of the plant breeding, it is only the increasing the productivity or the yield, right? Now, how the yield would be increased? When we are going for the creation of new varieties, and when those new varieties are further having improved agronomic traits, then they play a major factor in increasing the food production. But while we are going for the increase of yield, the very important component of the yield is its quality. Although if we are focusing on increasing the yield, say from billions we have increased the yield to the millions, say from grams we have increased the yield to the to the kilograms. But along with the yield, that yield will be further taken by the farmers, by the consumers or not. This in turn is controlled by its another parameter, that means the quality. That means we can define the quality as a degree of excellence. See, we are not to focus on increasing the Although the very immediate objective of plant breeding, very immediate objective of the plant breeding is to focus on the increased productivity or say yield. But this productivity or the amount of the yield that is increased, this in turn will be taken by the consumers. This in turn will be taken by the consumers or will be consumed by the farmers only if this yield is having good quality, right? So despite of the yield, there the is very essential component of economic yield is its quality. So we can define the quality. Quality is what? Quality is a degree of excellence. But when we talk for the a crop produced, then quality is the degree of excellence for a specific use or to serve specific purpose. That means when we're talking for the quality in terms of crop plants, then that quality is related to specific purpose only, right? So we can precise the meaning of quality, and this precise meaning of quality it vary depending upon the crop, depending upon its proposed and implied use, depending upon human needs and available technology. So the meaning of quality vary depending upon these four factors. This was all introduction about the say quality. Now we will discuss the quality traits. Quality traits means what? A character that defines some aspects of produced quality. Any character that is defining the quality of the produce that is produced is said to be quality trait. And the various quality traits are defined into, these are further classified into 5 categories. Very first quality trait is determined in terms of its morphology, then in terms of organoleptic, then nutrition, then biologic parameters and other quality traits. So quality traits are further classified into 5 broad groups. So let's discuss what is meant by morphological quality trait. As name itself is indicating, morphological means which are visible. That means we are talking here about all about the morphology. So the features are as these traits are related to produce appearance. These are mainly when we talk for the appearance, what all we discuss in the appearance. We discuss about the size, we discuss about the color, and very important thing about morphological quality traits, they are easily observable. That means you can see them very smoothly, and they play an important part because these are easily observable. So since the size and color of the produce can be easily observed, so it is playing an important part in determining consumer acceptance of the produce, because consumer will be directly say if this is the produce, and since we are talking about the morphological feature, so this in turn, this can be directly observed by the consumer, and when this is directly observed by the consumer, so this morphological feature is playing a very important role whether consumer will accept this product or will not accept this product, and whether it will accept this product or not accept this product, this in turn is concerned with the size and color of the produce. Why immediate decision is taken by the consumer? The reason is very clear, because this trait is under the category of morphological trait, which can be easily observed. So this is the reason, it helps in determining that it is helping the consumer to take the immediate decision whether to have this product or whether to not have this product. So quality must be like that, like its morphological features are very good in the nature. Second is all about organoleptic quality traits, and these include the features which are concerned with palatability of the produce. Palatability means Within the palatability, we talk for the taste, aroma, smell, juiciness, softness. You must have seen while you go for the purchasing fruits, then you ask shopkeepers to give you a certain part of the fruit, so that you could check its taste, smell, juiciness, softness, etc. These are said to be organoleptic quality traits. And even though if you have produced a very high yield, but the yield is no more having organoleptic quality traits, then again it will not be accepted by the consumer. So, uh, but these palatability features, they are also easily detected, and this is the reason they also play very important role in influencing the consumer preferences. That means whenever we are talking for the quality, quality is directly related to the consumer preferences. The next is nutritional quality traits. Nutritional, as the name itself is indicating, nutritional means now when we say for the quality, quality should determine the value of produce. That means quality should be of a parameter that it is providing whenever it is consumed by human or animal. It gives a balanced nutrition to the, it provides balanced nutrition to the human being or to the animal. And these nutritional qualities include characteristics like protein content and quality, oil content and quality, vitamin content, mineral content, and also the presence of anti-nutritional factors, because it is not always a particular yield, a particular character. When we talk for the nutrition, then it is going to have all the positive features only. Sometimes along with the good mineral content, we used to have anti-minerals factors also, which are available within the trait. So we need to see every parameter, and that every parameter needs to be analyzed very carefully. These traits are not easily appreciated by the consumer and farmers, but they are of very important value in determining the human and animal health. This is said because nutritional, it's not on the same day when you are going to have a particular nutrient you are going to consume, it is going to give you result on the same day. No, it doesn't happen, it never happened. Whatever nutrition you consume for maintaining your health, the result of that particular nutrition, they come after a certain days. For example, you are not growing up fast, so you visit the doctor and doctor suggests to you have a certain protein powder protein powder that is to be added into the lukewarm milk, and then it is to be obtained. So it is not going to be just the very first day you have milk that into which is added the protein powder, and very next day when you wake up, then you see you have a 1 inch height increased. This 1 inch height results, you will obtain the results, but the results are not very immediately produced. Results will be produced at a certain period. This 1 inch height, after the consumption of protein powder, it won't be achieved in within the night. It will be achieved, but it will take certain days, and those number of the days may be 20, it may be 1 month, or it may be 2 months. So this is the reason these traits are not appreciated by the consumers, because consumers know that here we have to wait for a certain period to see what results are coming out. But and so this is only the reason it is not appreciated by the consumer and farmers. Whereas when we were talking for the earlier two parameters, they were very easily appreciated, because the results were on the spontaneous results were being obtained. And here, since spontaneous results are not given out, so this is the reason these are not easily appreciated by consumers and by farmers. But they are important. Of course, these are nutritional quality traits, so they are definitely going to be important in determining the human and animal health. So this is about nutritional quality traits. The next is biological quality traits. Biological quality traits are the traits which defines its actual usefulness only when they are consumed by the experiment animals, right? Because see, whatever product that is produced, it is not whatever product is produced, it will be easily given to the directly, it will be sold into the market. No, that means that directly whatever breeding procedures will be done for improving the quality of a particular species. Okay, we think that those breeding approaches are successful, but once breeding approaches are successful in terms of quality trait, even then that particular variety won't be made directly available for selling purposes in the market. Very first they are experimented. We need to see that the quality traits we were looking after are exactly the ones or not, and this in turn can be estimated by introducing these good quality traits, or by introducing these improved quality traits into the experimental animals. And after they are consumed by the experimental animals, then their usefulness to the human is usually predicted. For example, these biological quality traits include protein efficiency ratio, biological value, body weight gain, etc. These traits are again not obvious to the consumer because they are again time consuming, so they are not obvious to the consumer, but are extremely valuable in terms of determination of quality of produce for human or animal consumption, right? The next is other quality traits. What are the other quality traits? These are the traits other than morphological, other than nutritional, other than biological, other than organoleptic. And these traits are won't be including any trait that is discussed in the above categories. Here simply we talk about a cooking quality of rice, keeping quality of fruits and vegetables, and fiber strength of the cotton. Many of the traits which are present in this group are of prime importance, and these are helpful in determining the usefulness of the concerned produce. I mean to say, these parameters won't be involving the any method that is in terms of that is present in terms of the first four factors, but it is of prime importance because it is again utilized in determining the usefulness of the concerned produce. So these all are the quality traits. Now we will talk from where these quality traits they can be obtained. That means we are to talk about sources for quality traits. When we talk for the sources of for quality traits, then quality traits can be obtained from a cultivated variety. Quality traits can be obtained from a germplasm line, from a mutant, from a somaclonal variant, from a wild relative, or a transgene. Very first that we will discuss about the source is about a cultivated variety. What is said to be the cultivated variety? A variety that is in progress, the variety that is cultivated is said to be cultivated variety. This cultivated variety, it acts as an most preferred source for a quality because of the reason this reason because they are easily utilized in the breeding programs, so these are the most preferred sources for equality. Because after all we are to focus on the breeding procedures only, right? So when we are to talk for the breeding procedure, in the breeding procedure mostly we try to have the varieties which are the cultivated ones. So the very important feature of cultivated variety is like these are the most preferred source, that whenever you are to breed any of your variety in terms of the quality, then you must focus on the cultivated varieties only. And the reason behind you must focus on the cultivated varieties is that they are easily utilized in the breeding programs. But if, see, I said when you are to breed, for a parameter that is said to be quality, then I said a breeder needs to focus on cultivated varieties. You are focusing on the cultivated, or say breeder is focusing on the cultivated varieties just because of the reason it is having that particular quality which actually we want to be present in our varieties. But if I say don't say quality, that means cultivated varieties have the characteristic that is important enough to improve the quality of a species. But what to do if a trait is not present? If the trait is not present, then breeder needs to look else somewhere for the trait. Then in that case, in spite of the utilization of the cultivated varieties, in spite of the utilization of cultivated varieties, breeder to look after the another approach so that this quality or whatever the platform we had, that is whatever quality whatever species we were looking after, the improvement of the quality that could be improved. This is cultivated variety. Again I repeat, a variety which is utilized in breeding programs, but only utilized when it is having the traits that are available for improving the quality. But if trait is not available, in that case breeder needs to look elsewhere for the trait. Now I will discuss the next source of for quality trait, and that is germplasm line. Here quality trait not available in the cultivated varieties. That means the characteristic that we were looking after in the cultivated variety, if it is not available, then in that case it may be found. We said in that case breeder needs to look elsewhere. But elsewhere where he needs to look? He or she can look after the germplasm line. And right, so this is a germplasm line. Germplasm, I have already told you in plant genetic resources, that is, it is the sum total of all the genes which are available for a particular species. So if a particular trait is not being found in the cultivated variety, then a breeder can look after in the germplasm for making that trait available. The next is mutant. Mutant is what? Mutant is the case. Mutant is the case where many quality traits, and when we talk for the many quality traits, we are basically talking for protein and quality traits. Within the mutant, the traits are contributed within the mutant, or the traits are present in the mutant, but those contributions are the results of spontaneous and induced mutations. Spontaneous means the mutations which are taking place naturally, and when we say induced mutation, in that case we are using the physical or chemical mutagens for creating the new changes, right? Next is somaclonal variant. Somaclonal variant may sometimes show an improvement in quality trait. For example, somaclonal variations are what? Like we are growing the cell or tissue under the artificial conditions. Here we had a cell or tissue of the plant, and we artificially prepared a media, and we allowed it to grow this cell or tissue onto this media. Under the artificial conditions, when there will be appropriate presence of the temperature, humidity, and light conditions, this cell that is present onto it, it grows and it gets converted into a plant. But while its development, certain variations takes place. While it is developing under the artificial conditions, certain, very important, certain variations takes place, and these variations are only the variations which are said to be somaclonal variations. And the result of somaclonal variation is going to be a somaclonal variant. And this in turn is also important in improving the quality trait. For example, somaclonal variant of sweet potato had deeper and more stable root color, and these qualities are appreciated by the consumer. As a result, these are preferred by the consumer, and this somaclonal variant of sweet potato with a deeper and stable root color is now called as a new variety, that is scarlet. This is called as scarlet. So when we are not going for the natural inductions of the variations, and variations can be induced as a result of tissue culture, and those variations are said to be somaclonal variations, and the variant that is developed is said to be somaclonal variant, right? Next is wild relative. We have seen many examples where genes for improved quality are contributed by wild relative. But one of the problem when we are considering wild relative as a source is the characteristic, that means a quality trait, is not expressed in the wild species. Quality trait is not expressed in wild species, but it is detected only in segregants recovered after its cross with the cultivated variety. I mean to say, we know that the particular wild variety is existing, and that wild relative variety is perfectly suitable for improving the quality of the for improving the genes for the quality. But one of the drawback when we consider wild relative as a source for quality trait, very one drawback is that sometimes the quality trait is not it is present, but it is not expressing itself when it is present within the wild relative. But when wild relative is allowed to cross with, this is wild relative. But when wild relative is allowed to cross with, say, wild species, it is allowed to cross with wild species, then at that particular time, we could see this quality trait. At that time, quality trait, it becomes observable. Quality trait at that time becomes observable. This is about a source for quality trait, and here the source is wild relative. Now this trait, because it is no more appearing in the initial stages, but when it is allowed to cross with the wild species, then at certain segregants, it could be seen. So this is the reason it is said to be latent traits. That means the traits which are observed in the later phases. It is said to be latent trait. Now, quality is clear to all. Quality traits are clear to all. And sources for quality traits are also clear to all. So we can easily go for breeding, and here these are the breeding approaches by which we could go for improving of quality. And here the breeding approaches are further classified into 5 categories. Very first is the evaluation of germplasm, then mutagenesis, then hybridization, then we have interspecific hybridization, and at the end we have somaclonal variation. Very first approach, these are the breeding approaches, and within the breeding approaches, we must be ensured that whatever breeding approaches we are using the breeding approaches for the improvement of quality traits. And again, I focus the improvement of quality traits. Here, we will discuss only for, the protein quality. So here is the very first breeding approach, that is screening of germplasm. What is screening of germplasm? Because we know germplasm, that means we call it as a gene pool where there are available all the genes which are present for a particular species. See, if this is the germplasm, and we need to screen this germplasm, and we are to screen the germplasm including, very important, including cultivated varieties. Because we have already said, like before we need to step towards the germplasm a source, we already have the quality traits which are present in the cultivated varieties. So we must screen the germplasm, but the germplasm must include the cultivated varieties also. And when we go for the screening, this screening yields a source for quality trait. But such traits that are not obvious. That means we are talking here about chemical composition, right? So here screening is done, and screening for the characteristic is done, for those characteristic is done which yields a source for quality trait. For example, chemical composition. Here high lysine germplasm lines of sorghum, they are screened. But it is not an easy task. Calling for the screening or saying performing the screening is not an easy task. It seems to be an easy task when we say we have to go for the screening of germplasm. But otherwise, when breeder actually needs to perform the screening process, it is not an easy effort. And these breeding efforts many breeding efforts are required to combine the quality trait with good agronomic features. Because why we need to combine breeding efforts along with the screening, because when we talk for the germplasm and when we talk for the features which are present in the germplasm, they are sometimes expected to be inferior means they are not going to represent itself. Inferior, superior means they are going to represent itself. But when we screen the germplasm and say, for fortunately if we had a good quality feature with us, then that good quality feature requires further breeding efforts, because the characteristics which are found in the germplasm, they are of inferior characteristic. Next is mutagenesis. In mutagenesis, sometimes neither the desired quality trait is neither present in the, sometimes what happens, quality trait is neither present in it is neither present in cultivated variety, neither it is present in the germplasm. Then what to do? If we are to have the quality trait with us, then what is another parameter that we need to look after? Then in that particular cases, the third breeding approach is mutagenesis, where a desired quality of trait, it can be obtained as a result of spontaneous or induced mutations. But when we go for the mutations, there come certain problems in the mutants, like the mutants have poorly filled grain and they give lower yield. Therefore, this is the reason again, like we were combining breeding efforts along with the germplasm, here also we need to use breeding programs we need to use the breeding programs so that we could develop high lysine, high yield, and superior varieties. And another problem while we go for the mutagenesis is it is not always whenever we induce a particular mutation, it is always going to give the desirable features only. Sometimes undesirable features are also present along with the desirable quality trait, and very important, these undesirable features are to be removed. They are not to be kept along with the quality traits, they are to be removed. Third is hybridization. Hybridization is the most widely used breeding approach. That is utilized in turn to develop high yielding varieties with desirable quality traits. But here, when we go for the breeding programs, then two parents are crossed among each other, and then we have a F1 generation where progeny is produced. Then from this progeny we have another generation F2, and so up to these number of generations may vary from one methodology to the another methodology, right? Here, what breeding methods are to be used to handle this? Okay, we have done the hybridization, but the generations which are produced after the F1, they are said to be segregating generations. Okay, we are performing hybridization, and obviously as a result of the hybridization, we are going to have a number of generations which are said to be segregating generations. But have you any thought like how these segregating generations are to be handled? Is there any idea like how these segregating generations are to be handled? But since we don't have any idea, we must be much familiar like when we are to handle these segregating generations, the handling of the segregating generations required certain breeding methods, and those breeding methods are mainly dependent upon the types of parents which are involved in the cross. Very important. That means in the hybridization, what we are going after? We are going after the crossing of two parents, right? Once the two parents are crossed, they are going to give you progeny that is said to be F1. From the F1 progeny, depending upon your motto, we used to adopt pedigree, back cross, any methodology can be looked after, and then it is going to give results to number of segregating generations. The number of segregation generation, it in turns is dependent upon what methodology you are using for the handling of segregating generation. This is one way, but obviously you need to handle the segregating generation to obtain the result. Which methodology is to be utilized for handling of the segregating generation? This in turn is dependent upon the parents which are crossed together. I will discuss it more deeply. Say, if both the parents are of a nature that they gives higher yield, but that higher yield character is again combined with good agronomic features, then in that particular case, we are going to utilize pedigree method for the handling of segregating generations. But in case any of the one parent has inferior agronomic features, then we are going to use backcross scheme, and the number of the backcrosses that can be done are either 2 or 3 in the number, and this will be the most appropriate method for handling of segregating generation if any of the one parent has inferior agronomic features. So this is about hybridization. Next is Somoglonal Variation. Next is Intraspecific Hybridization. Intraspecific means where species different. Wild relatives, they often contribute quality genes. Quality genes, that means a fruit color, fruit size, provitamin, and so on. Because wild relatives, they often contribute to quality genes, so this is the reason hybridization program is quite demanding. But along with the demand, it presents several problems also. So in any case, high quality line that is derived from such crosses that will be useful. Basically what we are doing, any high quality, because when we go for the interspecific hybridization, as I said, interspecific hybridization, when it is done in terms of wild relatives, it is also responsible for its contribution in terms of quality genes. When it is giving quality genes, then obviously it is quite demanding. But there are many problems also. So when we see its positive prospect, then because they are contributing to the quality genes, so these wild relatives are directly used as the parents. They can be used as parents in the hybridization program, and but they are not to be very important. But they are not to be directly released as varieties. They are to be utilized as parents, but they are not to be released as the variety, right? Next is somoglonal variation. This I have already told you. When we are growing the plant, we have a cell. We had a say leaf. Say we had this leaf. Then we had this cellular part of the leaf. On the other hand, we prepared a media that will provide all the supplement to this cell. This is the media. So this cell is when placed over the media, and when all the conditions, artificial conditions are maintained, say temperature, light, humidity, then because all the nourishment is available in this media, so this leaf will start growing into the plant. Further growth hormones will also be supplied to this media. Then as a result, what happens, this from this tissue arises the roots, and from this tissue arises the shoots, and it starts bearing the leaves also. This is growth of the plant under artificial conditions, or we can say these are tissue cultured raised plants. But while this growth of the plant is taking place from this particular cell, when the growth is taking place from this particular cell, what happens? Certain variations are introduced into this cell while it develops. And those variations which are introduced into its genetic content, that genetic variations are actually said to be the somaclonal variations. So genetic variations which are these are the genetic variations which are present in these tissue cultured raised plants are said to be somaclonal variations, but these somaclonal variations have been exploited. That means they have been maximally utilized. Maximally they are being utilized for the crop improvement. And like I discussed in the wild relative, where they are giving important features, they have the important features for the quality traits, but they were not being directly released as a variety. Here somaclonal variants, they have the advantage over the wild relative that they can be released as commercial varieties, right? For example, scarlet variety I have discussed a few slides back also. I discussed the scarlet variety of sweet potato. It has improved and more stable root color. So this is a result of somaclonal variations only. Modern technology. So now is the genetic engineering, and when we say genetic engineering, then in the genetic engineering, we basically talk for recombinant DNA technology. Genetic engineering basically involves recombinant DNA technology, recombinant DNA technology. Recombinant, you could understand. Recombinant means some two things are being combined together, and then when say I will change the color, say this is the one and this is another, then as a result both are when going to join together, this is said to be recombinant, right? This genetic engineering, before I actually enter into this genetic engineering, let's go through recombinant DNA technology. Actually, what happens in the recombinant DNA technology, we used to have source of the DNA. That source, here we will talk for the quality trait, and the quality trait source can be any source, wild, it can be germplasm, it can be It can be any that we have discussed in 5 or 6 varieties we have discussed. Any source of quality trait is selected. This is a genomic content that is isolated from a particular variety that is actually representing the source for good quality trait. And on the other hand, we had a vector that is having undesired gene. This source is having desired gene, say it is present here. Desired gene or say quality trait, right? We need an enzyme. These two are nucleic acid. So these both are DNA. So we need an enzyme that cuts this DNA, and as a result, this significant portion of the genomic content could be isolated, and from here, undesired portion of the gene could be removed. So we had the enzyme restriction. We had the enzyme restriction endonucleases. Restriction endonucleases. The restriction means it will restrict, it will cut. Endo means on the inner side of the DNA. Nucleases means an enzyme that acts on the nucleic acid, right? So this is the DNA. So it is acting upon the inner. It will not be acting on the ends, but it will be acting anywhere within the DNA. That is said to be restriction endonucleases. As a result, we have removed from here undesired gene, and on the other hand, this interesting gene, that means this required gene is also isolated. Now both of this gene will be combined here. So because see, this is also genetic content, this is also genetic content, because two genetic content, that means two DNA, they are combined together. So this is said to be recombinant DNA technology. And now actually we cannot directly incorporate, in certain cases we cannot directly incorporate this desired gene segment into our crop variety. So it is before the direct transfer or the direct introduction of this desired gene into the plant species required, it is entered into a vector, and that vector is useful. That vector is used for transferring this desired gene into the plant. So it will be now transferred into the host, say E. coli. E. coli is having its own genetic material, and when this will enter here, it will also have the desired gene. Now it will use the genetic machinery of the host, and it will start multiplying its number of copies. So this is the one of the ways how we can transfer our desired gene to the host cell. This is all about recombinant technology. This is said to be recombinant DNA technology. When we talk for the genetic engineering, we basically stress over recombinant DNA technology. And when I say recombinant, its abbreviation form is small r DNA, that stands for deoxyribonucleic acid technology, because all we are working over the DNA. So this is the reason it is said to be DNA technology. So genetic engineering is what? Genetic engineering is actually involving the introduction of a gene by the technique of recombinant DNA technology and genetic transformation. Now, see here one term, transformation, is to be utilized. Transformation. What is the transformation? See, this is recombinant DNA. If this recombinant DNA is uptaken by the host, very important. If the recombinant DNA is uptaken, that means it enters within the host, then we call it as transformation. Then we call it as transformation. So genetic engineering is involving two processes. Very first is the recombinant DNA technology, and second it is involving genetic transformation, because what is being uptaken by the host, DNA is being uptaken by the host, so this is the reason it is said to be genetic transformation. So genetic engineering is all about the technologies, about the techniques that involves recombinant DNA technology and that is involving the genetic transformation. When we go for the genetic engineering, its advantages are like one can use this genetic engineering for the improving the certain quality traits like protein, like oil, keeping qualities of tomato, flower color in case of petunias, etc. Because this is the desired gene. Suppose this host was no more producing proteins of good quality. Then we isolated a gene that was actually responsible for producing protein of good quality. We inserted into the vector, and that vector was later on because it could not be directly transferred into the host. So we used a vector, that means a mediator, that in turn transferred this desired gene into the host, right? So this is a way by due to which genetic engineering could be utilized to improve the quality traits like protein, oil qualities, etc. But within the genetic engineering also some examples of these developments are also there. Very first is modification of seed protein quality, then suppression of endogenous genes, then biochemical production. So within the genetic engineering, developments can be done by following either of these parameters. That means in the very first parameters, we modified means we need not to transfer the desired gene into the host. But the gene that is already present in the host and is not giving a good protein quality trait, that in turn needs to be modified. Rather than introducing new desired gene into the host, its own gene is to be modified, that is said to be modification. Sometimes what is happening within the host, there used to be the presence of certain genes which does not allow the good expression of quality trait for protein. So what we need to do at that particular situation, at that particular scenario, we need to suppress that particular gene which is not allowing the trait to represent its good quality. And third, we can go for the biochemical production, right? So these three are the examples by which development could be done under the genetic engineering. I will be discussing all these. I will be discussing all three examples in the next lecture. Now see, while one goes for the breeding, and once the breeding is being done for improved quality, then one must remember, if the variety is being improved, then along with the improvement in the characteristic, that means in the quality, that improved quality should not be at a cost of yield. That means if we are going for following of the procedure that is resulting into the improvement of quality, but there should not be any proportionality of quality with the cost. In the simple way, I could say, if the quality is being improved, then it should not result into the increase in the cost. Because if this is being done, it is not again going to be consumed by the consumers and the farmers. Because the very first preference of the consumers and farmers used to be, they want the availability of improved quality species in the market, but they also want that improved quality must be available at a very lower cost or at the cost that is easily in their access, right? Secondly, we should consider that when we are going for the breeding for quality, it should It should save the economic interest. This, when we talk for the cost of the yield, we are talking for the economic interest of both consumer and farmers. Very first is improving the quality should not improve the cost, should not increase the cost. And while we are going for the breeding for quality, one must consider into the mind that economic, that means when we talk for the money matters, that economic matters must be in the favor of consumers and farmers, so that it is easily available to them and they can easily consume it. Very important again, when we are improving the quality, right? When we are improving the quality, one thing that should be considered, okay, we have improved the quality, but it should not be like while we were improving the quality, that was high, giving high yield and stability, but when we utilize that improved quality after a certain period of time, that is no more giving the high yield and it is no more stable. It should not be like, its features should not change with season to season or with time to time. There should not be any variations in the characteristics. That means I would simply say the quality, the improved quality must have high and stable yields and good agronomic features also. But before we talk for these all big considerations, objective of the breeder must be very clear. He must be knowing what is to be done and what is not to be done, what is to be introduced and what is not to be introduced, what is going to be beneficial and what is not going to increase the cost of the yield. That means before we proceed for any breeding methodology, the clear-cut objectives of the breeder, they must be present, right? Because here all we are talking about the breeding procedure for the quality, and quality, when I say, I'm talking for only protein quality, and my quality stress is no more on the oil quality, just I'm stressing on the protein quality. Whenever we are looking after the breeding parameters for increasing the quality, then when we improve the quality, and if somehow that quality factor is going to diminish, that is, it is going to be destroyed, then breeder must be familiar with, if proper quality is not given, then what problems are going to be present in the human, right? So along with the improvement of nutritional quality, breeder must be familiar with the problems of human nutrition. Okay, and increased protein content is usually associated with a lower yield. So breeding effort, because when we increase the protein content, very important, when we increase the protein content, it is always associated with a lower yield. This is the negative association, and breeder must put all his efforts to break this negative association, because we are looking after the quality, but it is not like our motto is to decrease the yield. Quality and yield, they are both to be increased simultaneously or together, right? So these are the certain considerations in breeding. These are the other considerations, because when we are breeding for the quality, as I discussed in the last point, we are also stressing upon the nutrition. And if that particular nutrition is not being provided, we are looking after like a certain diseases they may be appearing. So breeder alone has not to work over the improvement of quality. Breeder must be having a continuous interactions with the nutritionist also. Okay, so that it will increase the rate of progress in breeding for nutritional quality. And very important, the success of breeding program for quality, improved quality, depends upon extent of genetic variability, heritability of the trait, availability of an analytical tool, and often success is often associated with undesirable effects also, because it is not like As I said, when we are going for the increase of protein content, we are always associated with the decrease of the yield. That is the negative and undesirable effect. So success is also dependent, and it is also including undesirable effects also. And the last genetic important consideration is, if in case genetic variation for a trait is not present in the germplasm, genetic variation for a trait is not present in the germplasm, then what to do? Then a suitable transgene could be used to bring about the desired alteration. As I discussed in the recombinant DNA technology, the desired gene that was being introduced into the vector, the desired gene that is said to be transgene. While I said no, very first step was the screening of, while you go for the breeding approaches, very first step was screening of breeding approaches. That means you were to go for the screening processes. But if genetic variation is not present in the germplasm, that means if you are not going to have any access in terms of screening also, then what is to be done? Then in that case, a particular transgene, it could be selected, and that could be utilized for carrying out the desired alteration in terms of quality. So these are the important considerations which a breeder must consider for improved quality. The next are the limitations. Limitations are what? Many traits are polygenic. Polygenic means they are producing many effects. And these are polygenic. That means many quality traits are produced by many genes. In that particular cases, when we are handling the segregating generation, in those particular cases, selection is somehow difficult. Secondly, quality traits further increase the list of traits. It further increase the list of traits a breeder has to select. And the last, it is always difficult to estimate and evaluate. Like it is when we say we talk for the quality, then it is very difficult to estimate. Quality traits are difficult to estimate and evaluate. So this was all about my today's lecture, where we discussed breeding approaches for quality improvement, right? So this was all about, and the one that is genetic engineering, where we are to discuss the three factors: modification, suppression, and biochemical production, that we will be discussing in the next lecture. So this was all from my side. If you have any queries, you can text me, you can call me, whatever mode of communication you feel comfortable, you can contact me. You can contact me through that particular mode. Thank you, and stay home and stay blessed.